there's no mean responsibility, I think, on my shoulders and my team's shoulders to make sure this is a success, and really want to share with you today what we've done so far. So we're going to change gear a little bit. It's our belief in our laboratory, but it's starting to become common knowledge that cancer is fundamentally a disease of the genome. And by that I mean that mistakes within cells, which we call mutations, can cause cancer. And clear cell sarcoma is no exception to this. And it has a very specific type of mistake or mutation, which we call a translocation. That's where 2 bits of DNA from 2 different regions of a chromosome get translocated to form an entirely new gene, not found anywhere else in nature apart from in this cancer. And that new gene forms a protein, right? This protein has cancer-causing properties. And for clear cell sarcoma, the most common variant of that abnormal cancer-causing gene is known as EWSR1, ATF1. Those are the names of the 2 genes. And actually EWSR1 stands for Ewing sarcoma receptor, which is also present in another type of rare sarcoma. That discovery was made in the 1970s. And since then, very little has been known about the genetics of this disease. So fundamentally, when we set out on this journey, we asked the question, what else could there be hidden in these genomes that could help us identify ways to target these tumors? And so the core objectives of the program was to first get a comprehensive readout of the genomic landscape of these tumors. And secondly, to improve our knowledge about disease progression and treatment resistance by understanding how these tumors grow and evolve, and also to look at how they might be different from one another. And then lastly, to try really hard, and I'll talk a little bit about this later, to apply these learnings to clinical trials with the aim of identifying new therapies. Now, what makes us unique or enables us to be able to do this, as I said earlier, is that we have one of the largest sarcoma services in Europe with really access to incredible expertise in cancer genomics, pathology and biomarkers, the understanding of evolution of cancer, and immuno-oncology. And primarily, bringing all of these together here at UCL, in discovery science, we can bring this to translational or translational applications in the clinic, train the next generation of scientists and clinicians, and really very important to us, and I was so glad it was important to the foundation as well, that this would be a global scientific resource. So all of the work that we're doing, all of the data that we're generating will not be buried here within UCL, but made available to the wider scientific and research community to be able to use. But we can't do this without people, right? And the talent pipeline that we've been growing has been incredible. We started in 2021, as you heard. The first port of call really for us, incredible to be able to appoint a technician to this role, who is Christy Davies, and you'll get a chance to meet him a bit later. I hope some of you have signed up to do the lab demonstration, which Christy will be leading. Christy was really instrumental here because, as you heard, these cancers are quite rare. Very few samples that are available, few and far between across the world. And he's been tireless in his efforts, not only within the UK, but also contacting multiple hospitals and laboratories across the world to create or generate one of the largest cohorts of soft tissue clear cell sarcoma that's available to date. For now, we're concentrating on the UK sample collection, but I'll speak to this a bit later. We have a much wider selection of cases that we're able to analyze through Christie's efforts. Through this program of work, we've been doing sequencing of these molecules within these cancers. We call this multi-omic sequencing, using different types of technologies. That creates an incredible amount of data. We needed somebody to analyze and interpret this data for us, and at that point we'd appointed a bioinformatician. And we're very delighted that Akanksha decided that she wanted to join our project and not some other very interesting projects in cancer, because I guess she understood the need. And I'll show you some of her work. But really also quite exciting for us in this here is that we've been expanding this talent pipeline. Young scientists and researchers all catalyzed around trying to understand a little bit more about clear cell sarcoma, and here 3 enthusiastic young clinical and non-clinical researchers who've joined the team, trying to understand a little bit more about clear cell sarcoma, and then more recently a master's student and a pathology trainee who is taking up the cudgels. So let me tell you a little bit about the sample cohort that we've got from the UK, right? They've come across from multiple centers, fresh frozen material. We have 38 primary tumors collected, 5 local recurrences, 12 lymph node metastases, and 5 distant metastases. And as you can see from the right-hand side, there are a wide array of technologies that has been applied across these samples to try and build the foundations, as Jane had said, these ingredients of the building blocks of what this cancer might be. It's already generated something like 20 terabytes of data. If you don't know what that means in terms of, or can envisage that, I was trying to do some back of the envelope calculations, and it's actually about 120 million volumes of War and Peace. That's a huge amount of data to try and work through that, to process it, to store it, catalog it, and we're able to do that here at UCL because of the computational infrastructure that we have that enables this sort of analysis. I'll just give you some headline figures about some of the clinical details of the patients that we have. We have a roughly even split, slightly more females than males in the patient cohort. The most common primary site of the disease is the ankle that we have, but you see there's a wide spectrum of other tissues, foot, hand, elbow, etc. We have no gastrointestinal cases yet. The tumor sizes varied, but actually many of them were diagnosed when they were quite large, so more than 5 centimeters in size. So at least half of the cases that we have are quite high stage disease. And smack bang on what we see from epidemiological studies, the vast majority of patients that we have in our cohort are between the ages of 20 to 40 years of age. How does clear cell sarcoma compare to other cancers? And what this map or graph shows you on the x-axis, a wide variety of different cancers. Now, one of the cancers that's often confused with clear cell sarcoma is malignant melanoma. That's sat here, right at the end of the spectrum. And what we're measuring is the numbers of these mistakes in the DNA or mutations that we have. And you can see that malignant melanoma has many of them. And that's partly intuitive. We've learned that actually those mutations or those mistakes are caused by damage from ultraviolet light. That causes them, this is why they have lots of these mutations. And actually that's what makes them amenable to treatment with immunotherapy drugs, right? Clear cell sarcoma is right down here, right? So it's really what people have been saying, and if you read the textbooks, say it's a quiet genome, not many mutations, right? And may suggest that they're not that amenable to immunotherapy drugs. However, the data that I'm going to show you now shows that it's not a quiet genome at all, right? This is a visualization that Akanksha's produced. It really helps us to try and look at the entire genome in perspective. We call it a circus plot. And on the outside of the circle are the chromosomes that are present within our cells, in this case, the cancer cells, going from 1 to 22, and then the X and Y chromosome. And the next circle down from that is the numbers of chromosomes across, the counts of those chromosomes across each of them. And you can see for the vast majority, there are 2, one from your mother and one from your father. But you can see that for some of these cancers or chromosomes, there are multiple copy number states, right? They're quite changed. And also, you find that some chromosomes are connected. So the classical mutation, as I described earlier, was the translocation between EWSR1 and this ATF1 gene, which is this little arc. But we found something really very, very interesting, not been described before in clear cell sarcoma, is that this arc doesn't just end there. It forms a loop, right? And that loop is very special. It's a specific type of genetic event known as chromoplexy. Creates this complexity in these genomes, and 25% of our patients show this. We don't know why yet. It's only being unraveled. It's quite common in prostate cancer, but never been shown in clear cell sarcoma before. So that was the first interesting bit of information that we've discovered from the sequencing. So that's the map there of that patient. Now that you're actually getting a crash course in whole genome sequencing here. I'm sure you're going to be able to go out and start to read these genomes on your own. I'll be out of a job. So what you can probably appreciate is that no 2 genomes are the same, right? They have the same fusion gene, that arc, but actually each of these cancers is quite different. Lots of different mutations, lots of complexity, which actually kind of explains why we're not able to get a handle on how to treat them, right? It may suggest that each patient needs their own personalized form of therapy or individualized to their particular genome. And that's quite difficult. There's some good news, however. is that there are some pathways that are shared between patients. And this is a bit of a complex graph, but I'll try and walk you through it. What we have at the bottom are the different patients. And what we have here on the y-axis are different pathways or different activities that are taking place within these cells that could potentially be targeted with therapies. And the more red the graph is, the more enriched that pathway is in that cancer sample, and the more blue it is, the less enriched it is. And you can already start to see hotspots, right? So here we have a group of patients who all have a similar sort of expression of a pathway that's involved with inflammatory signaling, meaning if you were to use immuno-oncology drugs, this might be the type of population of patients you want to use it on, right? And then you have other groups with other targetable mutations. So it really gives us a handle on which pathways we should be designing our drugs against. All of this data has really provided us with an incredible platform for research and investment in drug development. And we're really proud that we've won a very competitive scholarship from Sarcoma UK for Christy to transition from his technician role into now a PhD student, supervised by Rob Seller, who you'll get to meet a little later, Sandra Strauss, and collaborators at the University of Oxford to design an entirely new type of drug called a PROTAC for targeting clear cell sarcoma. We've also been able to get investment in terms of training for the next generation. And Gabby Rogan is a histopathology trainee who's joined our lab to try and learn a little bit more about these copy number changes in chromosomes that occur in clear cell sarcoma. It started to spread outside of the UK. So we now have a clinical fellowship funded by the University of Zurich for Dr. Simon Hayflyer, who's going to look at how these cells communicate with each other and try and find a way to disrupt it, which is really exciting. And then lastly here we have Dr. Dumila Miester, who's looking at gene regulation in clear cell sarcoma and funding that through his university. So we started from zero, and we've already started to build this capacity across the world. Also wanted to add that here in UCL, in a flagship project led by Adrian Flanagan, is starting to develop tools for better diagnosis of clear cell sarcoma using the digitized images that we have generated. In terms of educating the next generation, we have a PhD student in my lab and a friend of Ed's, Stephen Motley, who's put out an educational piece that's just been accepted for publication for other pathologists to learn about how to diagnose this disease. So we've really raised the profile of the research program. We've been across the world, London, Canada, Germany, Vienna, and our latest trip was in Dublin, where we presented the work that I'm showing you today. And really excited that both Akanksha and Christy presented their work on clear cell sarcoma, with Christy winning the Blue Ribbon Prize for the best poster presentation across the entire conference, really raising the profile of the research. So this meeting, together with all of this other work that I've shown you, has really inspired us. We have a huge number of priorities over the next 12 months. We really need to disseminate these findings to the research community, publications at conferences. We believe we started to lay the foundations down for designing drugs that target these tumors, and really to scope out other opportunities to partner with biotech and pharmaceutical companies to develop effective clinical trial strategies. And so with that short summary of the work that we're doing, I'm going to hand over to Sid, also a friend of Ed's, who will chair a session on a panel. Thanks, Sid.